Dear students, a very important area in bioinformatics is the study of protein structures. Proteins are essentially composed of amino acids which are brought together in a process of polymerization, and then these polymerized amino acids, they fold in order to give rise to three-dimensional proteins. A protein in a three-dimensional constellation can actually function by exposing certain portions of amino acid sequences for interaction. So it is a very important and critical area of research and study. Towards the start of this chapter, we are going to look at how each amino acid contributes to the structural property of the 3D protein. So to begin with, we're going to evaluate the properties of each amino acid individually. So the proteins, as I just mentioned, are assembled by ribosomes in the cellular cytoplasm, wherein one amino acid is brought after the other, and then this chain of amino acids, it is folded to form a 3D structure. The proteins and their properties that arrive after they have been folded are manifest only because of the properties inherent in their constituent amino acids. So simply put, the properties of the amino acids that construct the protein determine the property of the overall 3D structure. It is noteworthy that there may be some additional properties endowed upon the protein as a result of the formation of the 3D structure which was lagging in the amino acids. To begin the study of amino acids and their properties which contribute towards the formation of protein structures, we need to study each amino acid individually. There are 20 amino acids, as we know, and that each has a different chemical and physical property associated to it. When these properties are brought together after the process of polymerization, they give to a new set of properties that are imparted onto the protein. Here you see the structure of an amino acid with its side chain R given in the square box below. All the 20 different amino acids, they differ by their R group only. The rest of the elements, as shown here, the amine group and the carboxylic group, they are the same for all different amino acids. This is a very important point to consider. So any variation in the properties of the amino acids is essentially coming from the R group, as shown here. So if everything else is the same, then the R group will vary. Now the R group may comprise of different elements, different combinations of these elements, different lengths of these combinations, and so on and so forth. So towards understanding the properties of each amino acid, it is important to study the properties of the R groups. Here I show you the 20 different amino acids color-coded by their various properties. You can see in blue, the dark blue portion here, these are the basic amino acids. The ones shown here on the right side are the acidic amino acids. So basic and acidic amino acids together, they comprise the supercategory of amino acids called the charged amino acids. Secondly, the non-polar amino acids are shown here, while the polar amino acids are described here. So this set of amino acids has its own unique properties, and therefore by just looking at the type of the amino acid, you can essentially try to predict the property of the protein as well. You know that each amino acid has its own property, chemical, physical, structural, and so on and so forth. But when these amino acids come together after the process of polymerization, as shown here, you can see that water is given out and a peptide bond is formed. and the R groups, they are polymerized into a single protein. So therefore, different R groups, different amino acids with different properties, they come together in the form of a polymer which goes on to make the protein. This understanding is very important towards understanding the overall structural property of the proteins. So amino acids have properties such as polarity, charge states, and interactions with water, or simply hydrophobicity and hydrophilicity. And each of these properties taken together characterize the properties of the entire protein as well.